Class, thank you for joining me once again, where today I want to talk about a paper recently published, produced by some researchers in Sweden. This paper is published in Viruses, and it is entitled SARS-CoV-2 Spike Impairs DNA Damage and Inhibits VDJ Recombination in Vitro. So, as a public service, I thought we'd go through the too long didn't read version for you, help you interpret it a bit, and if anybody would like to make any commentary on anything that I may not have 100% correct here, please do that as well. The important thing is that people get good, robust, and correct information regarding what this paper is, what its meaning is, and where to go to from here, perhaps. Okay, so what did these researchers do? They took the spike protein, which has been identified from the SARS-CoV-2 virus. The SARS-CoV-2 virus, for those that don't know, is the one which is being linked with the COVID-19 disease. Okay, so what is the spike protein? It's one of the proteins that is produced by the viral packages when they infect cells, and it's a self-replicating protein that has various roles in the cycle of proliferation of the virus. Okay, so what have these researchers done? They have in vitro, meaning in a test tube. So this is not a study on living human beings in situ. This is not an in vivo study. This is in a test tube. They have taken some T cells and some B cells. They have taken some T cells and some B cells. T cells and B cells are part of the immune system. They are a vital part of the immune system. They are involved in detecting, binding to pathogenic organisms and assisting the immune system in trafficking the macrophage cells to these invaders for fighting and ultimate destruction of the invaders, hopefully. Right, what have they done? They have transfected the B cells and the T cells with spike protein, whole intact spike protein. So to transfect a cell in a test tube, you're going to have to inject the whole spike protein into the cell. So that's what they've done. So that's the first caveat. The spike protein from the SARS-CoV-2 virus is not known yet in vivo in living people to infect B cells or T cells. Okay, so that's the caveat. This is not known to occur. However, that does not mean it does not occur or cannot occur. It means it hasn't been seen happening yet. Okay, so we need to be very, very careful with what we say and how we say it. However, that's what these researchers did. They transfected the B cells and the T cells with whole intact spike protein. Right, what happened when they did that? Okay, the spike protein started to produce and proliferate a bunch of non-structural proteins which serve various roles in assisting the SARS-CoV-2 virus in doing what it does, basically, to keep it as simple and accessible as possible. These non-structural proteins are known as NSP1, 5, 9, 13, 14, and 16. Okay? These proteins were observed by these researchers to localize to the cell's nucleus. In other words, to go through the inner membrane of the nucleus and actually go into the central control center, the DNA center of the B and T cells. Okay? Once in there, the effect associated with those proteins being there was that there were several enzymes that are normally encoded for in the DNA control center of these cells. It's a message sent out to the cellular machinery to make these enzymes being involved in the repair of DNA that has become damaged, or indeed DNA that has been purposefully chopped up for slice and dice for recombination. One of the ways that B and T cells work is to do slice and dice, chop up the DNA, recombine it, put it back together, so that you have a whole bunch of different speciations, if you like, of B and T cells, which are involved in recognizing a specific invading pathogen. Okay, in this case, the SARS-CoV-2. What these non-structural proteins 1, 5, 9, 13, 14, and 16 are doing in the cell cytosol of these transfected cells and test tubes is causing these two repair enzymes known as NHEJ and HR. Parsimoniously, which enzyme is used to repair the DNA depends on what kind of damage has occurred to the DNA, the reason for that damage, in other words, also what phase of the cell cycle that cell is in. For example, in phase G1, the enzyme involved in the repair of DNA is NHEJ, and in the case of phases G2 and S, it's the HR enzyme. Great. Okay. Anyway, there's the non-specific proteins that have invaded that nucleus, that DNA control center of those cells, basically is vastly turning down the production of these repair enzymes. It would seem that would be a way of preventing the cell from generating these immune responses to this pathogen. So what we have here potentially is a spike protein which can have really HIV-like effects on the ability of the immune system to respond. Okay, very, very concerning. Okay, back to the earlier caveat that the spike protein is not known to infect B cells and T cells in a living human at this time. We haven't seen that happening. Again, to remind you, that does not mean it doesn't happen or cannot happen. It means it has not been seen happening. Okay, so that's what's going on there, basically. Furthermore, the other thing that's happening is that these cells that are transfected are producing a lot of other viral proteins and things in the cell cytosol and exuding those in exosomes, which can then go and transfect other cells of the body. So what we have here is a potentially very, very serious issue with having the SARS-CoV-2 spike in our systems. Now, there are two ways that you can get that SARS-CoV-2 spike into your system. One is you can be infected with the SARS-CoV-2 virus by coming into contact with somebody who is infected and who is currently shedding. That's one way. Another way is you can have it injected into your body in the form of a vaccine containing RNA instructions for the creation of an intact and whole spike protein, which in my understanding is exactly what is being done to millions and millions and millions as we speak. What are the possible ramifications of this? And I'm not saying that any of these things will occur. I'm saying, based on what I understand of what I've seen here and what I've read in this paper, here are some of the possibilities. First and foremost, obviously, immune dysfunction. the attenuation of the ability of the body to mount an immune response, an HIV-like problem. Okay, so that's the first one. The second possibility is all sorts of different carcinogenesis type effects. Cancer could be a real problem here, and other growth and proliferation dysfunctions within the cells involved in making up your body, basically. So what we have here is an extremely concerning issue, underpinned by an even more concerning report that I'm going to cover in my next video, where we look at a series of events leading up to the so-called approval of the Pfizer vaccine. So stick around for that. That'll be my next video. I hope this has been valuable. It's just a very short video to put the guts of this thing together for you. Please, if there are questions, let me have them underneath the video. And if there are any corrections, if there's anything I've got even remotely wrong there or I haven't explained well, let me know about that as well. It's very, very important that we do get this absolutely correct in terms of what this paper does say and what it does not say. I think I've been pretty clear. If you disagree for any reason, please do let me know what that is. Otherwise, I'll assume that everybody is happy with what I've said in terms of what it is this paper is inferring. Very concerning indeed. And I think that is something that we all need to be aware of as part of our decision-making processes at large. Okay, see you next time when we'll deal with the series of events as reported leading up to the Pfizer vaccine approval. Okay, see you then.